Mm. Sorry. Okay, let's get started. Okay, so welcome to the recap session or extra session for this course, NPTEL Computational Systems Biology. My name is Shomo and I'm KMRFTA, and this course is taught by Professor Karthik Raman of IIT Madras. Okay, so we will go through the entire course kind of on a on an overview kind of a thing. So we'll start from graph theory, then we'll move to different types of models, and then we'll move to different types of networks that we see and like how we can apply those models to those networks, and ultimately the finally stuff like HPA and its variations and how we can model using those techniques and find answers to certain questions. Okay, so let's get started. Okay, so initially we talked about graph theory concepts a little bit in the initial stages. So yeah, so a graph, by definition, you'll have a set of nodes and a set of edges, uh, uh, and these edges connecting between two nodes. And depending on the structure of the graph, you can have different types of graphs. So one of them, so the most common of them are undirected graphs, where you have nodes connected by edges, but there is no directionality to the edge that is being connected between two nodes. Then you can have directed graphs, something like this, so where you have these nodes and you have arrows pointing from one node to another, or edges connecting from one node to another, and uh, the, there is a direction associated with that connection. So it symbolizes the flow of information from one node to another. Then you can have different forms of these graphs. So directed and undirected are the most simpler ones. Then you can have something of a semantic graph, which is actually a pipeline kind of a thing. So this whole thing reads as farm, sheep is farm animal, something like that. So it's basically a connection between two nodes, and one after another, you have to add them together to get the whole information. So it's basically one big set of information, break it down into smaller ones. Then you have things like weighted graphs. So basically, you have the edge, the thickness of the edge, denoting the weight of the connection between two nodes. So if the darker the edge, the more tightly connected is the node to each other. So if you have an edge weight of zero point 0.1, it's given by a much more thinner line compared to an edge weight of 1, right? So yeah, these kind of graphs are mostly we see in our cases, we see uh, denoting strength of interaction between two elements. So for example, between a gene and a transcription factor, or between two different proteins binding together, or things like that. So mostly binding and interaction, things like these are shown by weighted graphs where the edge weight will denote the the, the affinity between these two elements, right? Then you have something of a mixed graph, so where you have edge weights as well. So basically, you have some edges are undirected, some edges have direction, some edges are weighted, some edges are unweighted. So you can have a mix of all of the situation. And then you have bipartite graph. So this is also a very uh, kind of a common example that we see in biological systems are these bipartite graphs. So in this case, you have two sets of nodes. So in this case, you have one set of node is 1, 2, 3, and the second set of nodes is 4, 5, 6, 7. So you have these two sets of nodes, but these nodes are not connected to each other. So 1, 2, and 3 are not connected to each other, but these connect to the other set of nodes. So between them, there is no connection. Between 1, 2, and 3, there is no connection. Between 4, 5, 6, and 7, there is no connection. But they connect to the opposite end of the other group of nodes. So 4 is connected to 2, then it is connected to 1 again. So uh, it basically does not connect with members of its own group, but connects to members of the opposite group. Then you have something like a multi-edge graph. So basically what it means is two nodes being connected by multiple edges, more than one edges. So it can mean different things, but yeah, so it, it, this is what we were talking about yesterday in terms of redundancy. So if you have multiple edges connecting two nodes, so in this case, some are direct connections. So basically, you can like you have two nodes and you have direct connections between these two nodes but you have multiple of those. So what these mean are basically redundancies in the system. So basically moving from one node to another or passing information from one node to another, you can travel through these multiple paths. Now, if one of the paths are deleted or mutated, in our case, those are the things that can happen. So you have other paths that you can follow, and the system will still be stable and it will not break down because of the deletion of the edge. Because you have redundancies in the system, your system becomes more robust. This is what we actually discussed last day. Right. And finally, this type of a graph, which is known as a hypergraph. So the edges actually originate from another edge. So, for example, you have these two nodes, 1 and 2, but the edges connecting 1 and 2 to 3, 4, and 5 are actually originating from another edge. So, yeah, so these are generally these different types of graphs that we face. Right. So coming to different parameters of of graphs. So yeah, we've discussed this intensively, but still let me go through it again. So first and the most basic parameter of a graph is its degree or connectivity. So uh, the degree of the connectivity is basically defined by K, which is the number of connections the node maintains with other nodes in the network. So it's basically how many edges does that node uh, uh, act as a sink or a source to. So basically how many edges are originating from the node and how many edges are getting are basically ending at the node. So these are the two types of edges that you can have, and you count them up together and you sum them up together for each node and you get the degree of the node. Now the degree of that is for every node, but for a network, what when we see is basically we take an average of all the degrees of all the nodes inside the network. So we find an average degree of the network. But remember, this the property is for a single node. It can be extrapolated to a network, but generally this property is of a single node only. Now, in this case, there can be two different propositions here. So one is an undirected network. So in that case, total number of connections is basically twice the number of sorry, no. Yeah. So basically, you are defining the number of links to other nodes. So in this case, um, for for the first node, node number one, it has two because two edges are there. For node two, it is three, 1, 2, and 3. For node 3, it is 2 again. and for node 4 it is 1. So the total number of links is basically one edge is connecting two nodes. So for when you're calculating degree of a single node, you have to divide it by 2. So that is where this half comes up. Yeah. So when you're calculating the degree, so you're calculating one edge for two nodes. So when you're calculating the degree per node, you need to divide it by 2. So that is where this half comes, and that's the sum of the total sum of all the degrees. So this is going to be something like this. Now, average degree, if you want to find, you divide it by n. So as we were talking about, we can also extrapolate this parameter for the whole network as well. So for the average degree, it's basically uh, divided by n. For directed networks, it's a little different because you have two types of degrees. One will be an in degree, so number of edges that are coming to the node, and one will be an out degree, so number of edges originating from the node. So the total degree will be the sum of the in and outs, and you can calculate the average degree separately for in degree and an out degree and so on. But uh, one thing to keep in mind in directed networks is you're not counting an edge twice sorry, you're not counting uh, uh, an edge twice for one single node. So here, as we are seeing, so two nodes are sharing one edge, but in this case, we are separating out in terms of defining the edges. So one is simply an in degree, and one is simply an out degree. So when I'm calculating an in degree for a uh, uh, for one node, I'm also automatically calculating the out degree of the other node uh, that that edge has originated from. So basically, the total sum of the degree is sum of in and out for a directed network. Now we have different parameters to define the type of network. Uh, just a second. Yeah. We define different paths and distances in networks because as a proxy to how much is actually one node far away from the other. And by that definition, we can understand a lot of properties of a network. So one of them is the characteristic path length, which is the number of edges in the shortest path between two vertices, meaning two nodes you want to travel from one node to another node. What is the shortest path between these two nodes, and how many edges are there if you have to cross that path? So that is what is known as the characteristic path length. It basically typically measures the separation between two vertices in a network, and it also represents a network's overall navigability, which is also intuitive in nature. Okay, so that is the characteristic path length. So it's just simply two nodes you're connecting and the number of edges that are connecting between these two nodes. Let you define something called a network diameter. So as the name suggests, so for a sphere or a circle, we define the diameter uh, as the longest possible line connecting two points in the circumference in a in a circle or a sphere. That is what a diameter is. So basically, two points which are farthest away from each other. What is the distance between them? So for the network as well, it's a similar concept. So you have two nodes which are very far, which are actually the farthest away from each other. If you compare the other nodes in the network, and you want to find what is the actual distance between these two nodes, that gives you the network diameter. So it is also uh, viewed as the length of the longest shortest path in the network. So basically, uh, between two nodes, if you calculate the shortest path, what is the longest among those? So that is what the network diameter gives you, which is intuitive in nature. So in general, the characteristic path length defines the navigability between two nodes, and actually the network diameter tells you how vast the network is in terms of one extreme end of one node to connecting to the other extreme end of the other node. Right? So here is an example. So basically, if you consider the shortest distance between 1 and 5, it's basically 1, 2, and 5, not 1, 2, 3, 4, and 5, right? So that is the shortest path. Again, 1 to 4, it's 1, 2, 3, 4, or 1, 2, 5, 4. It's the same. But if you want to calculate the diameter, you have to calculate the longest shortest path in the network between any two nodes. So the longest path between any two nodes will be 4 and 1, and the shortest path there is 1, 2, 3, and 4. So that is the network diameter that defines what is the distance between two extreme nodes in the network. Now we come to the concept of clustering coefficient. In simple words, the clustering coefficient of an undirected graph is basically the number of triangles in a graph. So the number of triangles is the smallest connected component, fully connected component to the graph. So you have three nodes and you have connection between all three. Okay, that's the shortest connected component that you can have, or the smallest connected component you can have in a graph. So the clustering coefficient is the measure of number of these small triangles or the small connected components in the graph, and it is given by the number of the formula is given by the number of triangles connected to node I by the number of triples centered around node I. So where the triple centered around node I is a set of two edges connected to node I. So the clustering coefficient as an average you can extrapolate to the whole network, where you can average out the clustering coefficient of each node and calculate for the whole network. Yeah, so basically, if you want to calculate the clustering coefficient, it's basically you calculate the number of so consider all these three graphs. So for on the right hand side, you do not have a full triangle connected to this node I, right? So uh, it's not possible to calculate. There is no clustering coefficient for this particular graph, so it's zero. For this one, you have one, two, and three of these triangles uh, connected to node I, and so basically the formula is given by 2L by k into k minus 1. So where L is the number of links between the neighbors of I in a tree, which is basically this is in KC2. So you have say for example k nodes centered around your uh, particular node of interest. So the number of possible clique is that shortest connected component of one particular triangle. So if you see the clustering coefficients of these graphs, so on the left you have the highest one, and on the right you have the lowest one. So intuitively you can understand what it means. So if your clustering coefficient is very high, then your connectedness of the graph at that module or that component is very high. So it means it is densely connected. If your clustering coefficient is less, then what happens is it basically it, it means less connections, and all possible connections are not there. So in this case, all the possible connections are there between these five nodes. But in this case, all the possible connections are not there. So only half the connections are there if you calculate. So that is why it becomes 1.5. And if you want to calculate the clustering coefficient of all the nodes together, so basically you have to calculate the for all the five, and then you add up. So in this case, since this is a fully connected graph, the clustering coefficient will always be 1, and these graphs will have lower. So it's an important metric which tells you basically how much connected the graph is or not. Okay, we come to another important parameter which is known as closeness centrality. So it, it, it basically means what it says, how close are basically two different nodes. Uh, it's it's defined as if the farness or the peripherality of a node is defined as the sum of its distance to all other nodes, and the closeness is defined as the inverse of the farness. So between two nodes, what are all the possible sum of so so if you have multiple paths connecting to different nodes, what you're doing is calculating the distance for all these paths, and you're adding them up, and then you're taking an inverse of that, which gives you the closeness. So if there is no path between vertex v and I, the total number of vertices used in the formula instead of the path length. So basically, if you do not have a direct path between no connected path between these two vertices, then the total number of vertices is used in the formula instead of the path length. So total number of vertices that are between these two nodes are calculated, not the distance. The more central a node is, the lower is its total distance to other nodes. So basically, if you intuitively you can think, so you're calculating, so you have one node and you have say 50 other nodes in the network. So you're calculating the distance of this one node to the rest of the 50 other nodes, and then summing it up, and then inversing is obviously. But if that no, if the closeness centrality that you're measuring of the node is very on the peripheral side of the network, the the value of closeness will be lower because the distance will be higher to all other nodes. But imagine if that node is in the center of the network and you have distances on the left and right, and the distance is actually less than what the node at the periphery will have. So it basically gives you how centrally located the node is. The more central the node is, the lower is its total distance to other nodes. And closeness can be regarded as a measure of how long it will take to spread information from that node to all other nodes in a sequential manner. I'm like going over all these topics one by one. If you have any doubts, please stop me and ask questions, by the way. Okay, okay. Next parameter we come to is called betweenness centrality. So it's basically, I'm not reading it out from here, I'm just giving you the intuition. So you have two nodes in the network, and you have a path connecting these two nodes, right? So what you want to measure is a third node present in between the paths of these two nodes. What you want to measure is how many of the so initially you started with two nodes, and say for example you have five paths connecting these two nodes, right? And you have a third node for which you want to measure the betweenness centrality. So what it actually means is you have to measure the number of the shortest paths between these two nodes, how many of them actually pass through the third node, right? So if you have say five shortest paths between the two nodes, and two of them pass through but a third node. So so the betweenness centrality of a third node will be 2 by 5. It's 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 actually that. So what it means is betweenness centrality of a node is is intuitively what it means is how many times it is in between two different nodes. How many times it is in the shortest path between two different nodes. How many times if you want to go from one node to another, you have to pass through that node. So in a systems biology perspective, or in a general system, if your betweenness centrality is high, that means if you want to move from any random node to any other random node, it is possible that you have to move through that node. So that actually becomes a very important component in the network, because that node is connecting a lot of shortest paths between two different nodes. So it actually becomes an important part of the component of the network, and if you disrupt it, the connections between the nodes might get disrupted. Right. So yeah, it's given by this. So betweenness centrality is this ratio of the uh, the total the number of so on the denominator you have the total number of shortest paths between two nodes S and T, and on the numerator you have the total number of paths that pass through another particular node V, of which you're measuring the betweenness centrality of, right? Okay. Now, the concept of random walk. So basically, it's uh, it's a stochastic or a random process which basically describes the path that consists of a succession of random steps on some mathematical space. So, for example, you in a given graph, you start in a node. That's your starting point, and then you select a neighbor at random. So neighbor is basically all the nodes that are connected directly to that node via some vertices or edges. And then you move to the selected neighbor, and then again you stop there and you see all the neighbors, and with some probability you move to another neighbor. So you do this, iterate over this, uh, repeat the whole process until termination condition is verified. A termination condition can be reaching of a certain node or traveling a certain distance or whatever it can be. And this random sequence of points selected in this way is called the random graph. And Random graphs, sorry, random walk of a graph. Sorry, random walks are very commonly used. Say, for example, in genome assembly, where so you have in genome assembly, what happens is you break down the DNA into small portions, and there are overlaps between these different portions of the DNA, which are known as schemers. Now, if you imagine that in a graph concept, so you have these schemers representing each of the node in a graph, and connection between two gamers means that they are having overlap. Now, if you want, if you only have this information, so you have all the nodes, so all the reads in the graph you have, and you also have the connection between two different reads where there is an overlap. Now, if you are asked to find the whole sequence of the genome from this information, a random walk of the graph is a very good idea to do. So you start at random at one node, and then you move to another node with some probability depending on the number of overlapping points that you have with another node, and then you go on and on, and then until you uh, reach a termination criteria, you go on and on, and then you can have the whole sequence. So a specialized version of this is an Eulerian walk, that is also a random walk in the graph, and you can have modifications of that, like random walk with restart. So basically, you reach reach a condition, you do not terminate the walk, you actually go back and start it again. So that can also happen. So it is a method that studies the network of vicinity of gene protein seeds to study their function. Sorry, it is based on the idea that nodes related to similar functions actually tend to be close to each other. In other applications, random walk is used to model different scenarios. For example, Brownian motion of dust particles. It has applications in statistical mechanics as well. And in computer science, it is used to model epidemic diffusion of information, generate random samples from a data set, and compute aggregate functions in complex sets as well. So it's a vast array of applications, but in our case, mostly in disease or genome assembly and things like this is used. Okay, now we come to a very important concept, specifically for our scenarios, is the centrality lethality hypothesis. So the idea is that if you have nodes that have very high connections, if you delete that node from the system is probably going to collapse, or in our case, the individual or the species is going to die, right? So the idea is that if you have a node that is highly connected, that means that is a very important node. So in our case, you can imagine it as them as proteins which are involved in a lot of processes, and those are like housekeeping proteins or housekeeping genes that are always required under a lot of processes. And if you by chance delete that gene or knock that gene out or remove that node from the network, the system will eventually die because you have removed a very important component of the system. So the hypothesis is basically protein with higher degree centrality are likely to be more lethal. That means proteins involved in more interactions are more likely to cause death when knocked off, which is kind of intuitive in itself, right? And another concept following this is basically pairwise connectivity index. It's basically quantifying how crucial an individual element is for sustaining communication ability between connected pairs of vertices in a network that is displayed as a directed graph. So you have the uh, yeah, so pairwise disconnectivity index uh, is this is the formula for that, where n naught is the total number of vertex pairs in the network that are connected by a path of any length in the network, and nv is the number of vertex pairs that remain connected following the removal of vertex v, and then you divide it by n naught. So basically the idea is, if you remove one node, how is the connectivity between two other nodes getting affected, and like what is the percent change of the connectivity you see after knocking or deleting a particular node. Okay, so finally, we saw all the kind of, not all the, but kind of the major parameters or centrality measures of networks. But our focus is to understand perturbations to the network, because in our case, the network is a biological system, and perturbation to the system can cause diseases. So our uh, focus should be on diseases, should be on network perturbations, and how we can find diseases through that. So cellular networks are generally robust against various perturbations because there are a lot of redundancies, as we were discussing, but it can be extremely fragile to specific ones. This trade-off between robustness and fragility can lead to this disease formation, and uh, this can happen when, for example, certain gene mutations or combinations, they can trigger kind of cascades of failures in gene regulatory network, which can lead to disease phenotypes. And you have diseases like Huntington's disease, cystic fibrosis, sickle cell anemia. These are like monogenic diseases. So basically, in one gene, this mutation has occurred, and you can find, and it's kind of easier to find than other multifactorial or polygenic diseases like cancer or diabetes or uh, Alzheimer's, Parkinson's, etc. These are polygenic diseases, and um, uh, like various number of combinations of mutations of other genes can cause this phenotype. So we can have a lot of variety of perturbations in the network. So, for example, one perturbation can cause a disease, but also like 50 different perturbations can contribute and cause a disease as well. So, yeah, we have to be careful about that. Okay, so then we come to the concept of community detection. So the idea is you have a very big network, and inside you want to find modules of the network which are highly connected with each other, but are less connected to the rest of the modules in the network. So you have a big network. Say, for example, you can divide the network into 5 modules. The idea is that if these are communities inside the network, so these 5 modules will have nodes that are connected to very highly connected to each other, but edges traveling from that community to another community will be very low. So yeah, this is a example from the kind of, this is an analogy to what I was saying. So basically, the idea is to the community detection is the problem of finding uh, natural divisions of a network into groups of vertices, which are known as these communities or clusters, such that there are many edges within groups and few edges between groups. So the idea is that these communities will have very densely connected nodes within each other, but the connections between nodes between one community and the other community will be very few. Okay, so there are different methods that you can use to find um, these communities inside the network. One of them is hierarchical clustering, uh, and for hierarchical clustering, is basically you're clustering the nodes based on how connected the nodes are and what is the distance between two nodes. uh, so basically, you start with one node, then you connect a second node if the distance is smaller than a certain cutoff. Then you connect the third node if the distance is smaller than a certain cutoff, and like that you make the whole community. And you break this loop after getting after like a few iterations, when the distance between a node inside the community and outside the community becomes greater than a certain cutoff. Now, that cutoff and the distance parameter is very important for the clustering purpose. So for that, you can go by very the simple uh, plus the simple distance matrix, which are basically Euclidean distances. But you have to keep in mind that Euclidean distances are mostly symmetric in nature. So basically, the distance between A to B and the distance between B to A is similar. But in case of the networks, it is specifically in case of directed networks, that is not the case. So the distance between one node to another node and the opposite of that from node A to node B and from node B to node A, the distance are not always going to be same because specifically in directed graphs. So you have other distance parameters there. So you can use actually cosine similarity measure, which actually is a dot product formula. You can use that. You can use something called as a entropy measure. So uh, yeah, you can use a lot of different parameters for the distance matrix. Right. So just I wanted to say this because Euclidean does not always work. Okay. Now we come to the another algorithm. So that was hierarchical clustering, which you can use for community detection. Another important algorithm, and actually a more popular algorithm for community detection, is the Garber-Newman algorithm. So it's an iterative process. Again, you have to do that. Um, it has to be an iterative process to find communities inside the network. But yeah, so at the beginning of every iteration, you compute the betweenness between two uh, betweenness of the edges in the graph and remove the edges with the largest betweenness. So basically, the idea is if you have a network, you find the node which has the highest betweenness in the graph. So what does the highest betweenness in the graph mean? That node is actually connected uh, is actually connecting a lot of other different nodes as the shortest paths are passing through that node. So a lot of different nodes are actually passing through the paths of a lot of different nodes are passing through that particular node. So the highest betweenness will actually node is actually serving as a connection between two different uh, set of nodes, if you can imagine like that. So the idea is you identify that node of highest betweenness and then you remove it. And then again, you do the same. You find what is the node having the highest betweenness, then you remove it and you do it iteratively until there are no more edges left in the network, right? Uh, and you calculate, so for each of these modules formed after you remove this particular the highest betweenness wala node, and after you remove that node and the communities that are formed, you calculate something called the modularity of these networks, of these small communities. And yeah, you stop the iteration. So your termination criteria is you repeat the iterations until there are no more edges, and the partition with the total modularity score, the highest total modularity score is actually the optimal partition. Uh, and you can stop this as well when uh, the total modularity score of an undivided community is greater than the modularity score of a divided community. So this is an example network where you have, I think, eight edges, eight, sorry, nine nodes, sorry, nine nodes inside the network. Now, if you follow Garber-Newman algorithm, you remove the edge with the highest between the centrality, which is going to be this E, and uh, the edges connecting E and F and edges connecting E and A. So that is the node with the highest between the centrality, and then you remove these two edges, and you are left with two different communities. Now you can consider these two communities and calculate the modularity score of these communities, and then again remove the node with the highest between the centrality, and then you again calculate the modularity on an iterative basis uh, until and unless you have exhausted all the edges inside the network, or you have actually your total modularity score is better for the previous iteration compared to the next iteration. Okay, as I was saying, modularity is the parameter that we are using to kind of define how good or bad the partition is in the network. So yeah, so uh, uh, Yeah, so I'll just read from here. Modularity measures the strength of the community partition by taking into account the degree distribution. And given a network of with m edges, the expected number of edges between two nodes I and j with degrees di and dj are given by di star dj by 2n. So this is actually the formula. So if you have m edges and two expected number of edges between two nodes I and j as di and dj. So this is the formula di star dj by 2n. Uh, for the uh, I'll go through. I, oh no, I actually went through a very good example for Garvan Newman when I was actually discussing this in class two, I think two or three. I You can actually look that up. That example is very good to understand the modularity and the Garvan Newman algorithm. Right. But the idea is that the true community structure in a network corresponds to a statistically surprising arrangement of edges, and that can be quantified by this element, which is the modularity. Uh, the modularity is up to a multiplicative constant, the number of edges falling within groups minus minus the expected number of edges in an equivalent network with edges placed at random. So the idea is uh, say you have 10 nodes in your network and you have certain connections between these 10 nodes. Now the idea is for modularity is, so after you partition, so the partition is there inside the node, and the modularity will measure the strength of the partition by taking into account this degree distribution. And how does it do it? So it compares your network to a random network of the same number of nodes and the same number of edges, but the edges placed at random. So if it happens that yours is a statistically significant network in which the edges are more close within that set of nodes compared to other nodes, so that means that is a good partition. So statistically significant partition. So uh, that is the thing the modularity takes advantage of. Right? Okay, now we come to something called as network motifs. So, what is a network motif? It's a subgraph or a subnetwork of the whole network, which actually occurs more frequently than other subgraphs in the network. So, you have a set of connected nodes inside the network, uh, and it occurs much more frequently than you would expect in network. So that is what is known as a network motif. So to identify a motif, so you first identify all the subgraphs of all the nodes in the network, and then you randomize the network while keeping the number of nodes, edges, and this degree distribution the same, and you find out all subgraphs that occurred significantly more frequently in the real network as compared to the randomized one. So the idea is if you have a certain number, a certain subgraph that is actually not getting created by random, and there is you find statistically significant evidence that that subgraph actually occurs much more in your network compared to a randomized version of the same network, you can call that actually a network motif. So we have different models to find. Sorry. You have different models to find network motifs. One is the configuration model, one is the edge switching model. I will not go into the models in details. We've studied these earlier, but yeah, this is the idea for network motifs. Just give me a second. I'll come back. Okay. Okay. So we've covered the network parts, the basic measures of a network, what all you can infer from a network, and how can divide a network into different components. Now, the idea is to how to use these networks in models of biological systems. So actually, this is a good uh, representation of modeling in systems biology. So you start with some prior knowledge of components, interactions, pathways, or networks. Then you develop a system of coupled ordinary differential equations, and you use parameters to better describe the equations. Then you basically do a simulation or parameter estimation, and then you understand information processing mechanisms. But at this position where you are determining the parameters and solving the equations, depending on different parameters, you'll get different results. Now that parameters needs to be um, well within a description of an experimental data or a or an extra or some sort of a hypothesis, right? So that estimation of parameter can be done from a experimental data, or use you can use a guesstimate parameter values based on the biology of the component. But all this actually require a prior information. So you start with a question, you do a simulation, you get the parameters. Do the parameters actually describe the system, or do you need a little bit more information on those? So depending on that, you do this is kind of an iterative process that goes on for until and unless you get a good model, and actually the model that describes your situation or condition in a better manner than before. Now, if you if you talk about biological modules, so in case of ODs, this is how we define an OD. So this is basically you have an element which can be a RNA, protein, molecule, DNA, whatever. So what you want to do is check the concentration of that element of that compound at T plus delta T compared to T. So this is how it is given. X of T plus delta T equal to xt plus k xt or into delta T. And if you take a limit, you get dxdt. So this is actually the differentiation formula, or the differential equation formula. So dxdt becomes kx. So uh, what it means is, so the power of x is actually telling you the order of the ODE. So this is actually first order. It is linear, and it is also autonomous because you could directly solve. This is first order linear autonomous ODE. It involves two quantities, which is the state variable is the x, which you are trying to determine, which is concentration of the compound, and k is a parameter. Uh, it does not describe an elementary component of the processes. It describes a reaction rate. That is how the concentration of X actually varies with time. Now, this parameter, K parameter, in most cases, in most models, you need to estimate separately. So there are various methods for that. You can statistically, there can be various methods to do that. One is maximum likelihood estimation and least square fitting. These are like common regression schemes. They can yield parameter values and their variance covariance matrix. But there is an assumption that the estimated parameters actually have a normal distribution. And when this assumption is not valid, Bayesian inference can be used, yielding a full parameter distribution. Uh, uh, yeah, so post regression you can do is testing a model's goodness of fit, determining which model among the competing ones actually fit the data, evaluating parameter determinability. So basically, you have different parameters, all of them might be equally likely. So what you do is put the parameters in a model and see how the data fits your model. That is the goodness of fit. And parameters in probabilistic model, they can be either inferred by indirect inference or Bayesian methods. In indirect inference, parameters are estimated by minimizing difference between intermediate statistics that characterize the simulated and experimental data. Is basically something of a least square thing. So you have a regression line, and then you have data points in and around the regression line. What you want to do is minimize the distance between these data points and the regression line. So that is one such method. Okay, so this part I'll go over quickly because again, there's not a lot of kind of examples that we have discussed on here, but these are actually a very important part of the algorithms that we use to understand systems biology. So evolutionary algorithm, these are basically based on the principles of evolution. So in general, you have a population. In the population, what a population means is basically a set of parameters that actually solve a particular equation. Now you can have uh, sets of these parameters together depending on your parameter estimation technique, and each of the set of parameters are known as a chromosome. So basically this is a bit array. This is the whole population. So uh, this is one set of parameters. This is one set of parameters. This is one set of parameters. This is one set of parameters, like this. So you have a set of parameters in a population, and one set of parameters is known as an individual chromosome, and one individual parameter in a chromosome is known as a gene. So these are like analogies to the biological system. Population is defined using the first generation or F1. This can be a set of genes, chromosomes, or other units of interest. So we are talking in terms of genes and chromosomes, but it can be something else as well. The most common technique is to use a bit array. So you use either 0 or 1. Uh, this is in case of your binary operators or binary models, but mostly Boolean models. So mostly um, it will be individual parameter values, not only always zeros and ones. Right? The algorithm goes as follows. So you have a population initiation set of parameter values. Then you have some sort of a fitness function, or basically uh, kind of a function that actually tells you how good are the parameters fitting to the model. So that's a fitness function. Then you do a crossover. So basically, you randomly change these parameters among different chromosomes, and then you mutate one or two of these parameters and see which of them actually is surviving the best, and then you put that into fitness calculation if it is better, uh, if it is fitter than the previous uh, set of uh, set of parameters. Right? So yeah, this is how the algorithm works. So you generate random population of n chromosomes. Sorry, generate random population of n chromosomes, evaluate the fitness of each chromosome X in the population. Then you create a new population by repeating uh, the following steps. First is a selection. Select two parent chromosomes from the population according to their fitness. Then you do crossover, change values. So crossover values between two different chromosomes. So for example, this chromosome and this chromosome, you can change different values, like in a crossover that actually happens uh, in uh, in cell cycles, right? So yeah, then you do a mutation. In biology, we understand mutation as change of base pairs in the DNA, but in this case, it's change of parameters. You switch from 1 to 0 or 0 to 1 randomly, and then you again calculate fitness function and say if the fitness of current set of solutions is better than the previous set of solutions. If not, you repeat the process again and again and again, and until and unless you get a set of parameters that actually have the best fit value according to your fitness function, you choose that. And that is what are genetic algorithms and evolutionary algorithms. Okay. Now we come to a very important component of our course actually. It's the metabolic networks. We have focused a lot on metabolic networks. So these are networks comprised of chemical reactions involving metabolites, catalytic proteins, and these metabolic networks pathways consist of successive reactions that uh, convert educts to products. And when it's the educt and the product are same, it forms a cycle. Right? Then you like a Krebs cycle or something like that. The anabolism and catabolism are like the two majorly used terms for metabolism. So anabolism is used for synthesis, catabolism is used for degradation. Uh, yeah, the network is represented with, with the edges representing the educts and products. Reactions are sorry. with the nodes representing the products and the substrates. The reactions are actually the edges connecting these products and substrates and so on. And for simulation, the knowledge of reaction kinetics is crucial because you need to know the rate of the reaction for it to be modeled well. Okay, and this is one example. I think this is mm, yeah. So you're creating, I think, tetrahydrofluoride from dihydrofluoride. So this is going to be one node. This is going to be one node, and the edge connecting these two nodes is actually the reaction converting dihydrofluoride to tetrahydrofluoride. Now, the idea is that you have to construct the metabolic network first and then do the analysis, right? There comes the concept of network reconstruction. So you compile the data, the enzymes, transport proteins from annotated genome sequences, like databases like Metasec or comprehensive microbial resource or anything like that, string or whatever, right? And then you uh, use functional reconstruction for phenotype simulation, requiring all proteins necessary for known organism behaviors. You can integrate genomic, biochemical, and phenotypic data for initial reconstruction. So basically, for reconstruction, you need to have a priori knowledge that is either from databases, either from experiments, or so on. So this is actually the metabolic network of our body. So I don't know if you can understand. This is the Krebs cycle here. Glycolysis is somewhere here, and so on. Lipid metabolism is this module, energy metabolism is this module, and so on and so forth. So it's a very complicated network as expected, but again, for our understanding, we will use different techniques. We use different techniques to understand the network. So the first important component of the metabolic network is the stoichiometric matrix, right? So you have a reaction. Excuse me. Yeah. So you have a reaction, which is here is A plus B giving 2C. Sorry. Yeah. So you have a reaction like this, A plus B giving 2C. So the stoichiometric coefficients for here for A and B are going to be -1 and -1 because A and B are getting used up, and C is getting produced, so it's a +2 there. So the three stoichiometric coefficients are going to be -1, -1, and +2. Uh, uh, Now, if you assume the metabolic network has R number of reactions, R1 to RR, then the reaction Ri operates with a velocity or the rate Vi. And then you collect individual reaction rates to a rate vector, which is the transpose of this V1 to Vr. And similarly, the concentration vector is also there, x1 to xt transpose. Now, for steady state approximation, this is the condition, dx dt equal to SV. So the change of uh, the change of rate of change of concentration of a substrate is zero is equal is given by product of the stoichiometric matrix and the velocity of the reaction S into V. At steady state, this dxdt becomes zero, so where SV also becomes zero. Right. Yeah. So the required production to equal consumption of each metabolite, which forces the reaction rates to be invariant over time. So that is what the steady state is about. The rate of product formation is equal to the uh, redu- uh, uh, is equal to the rate of product degradation. So it's steady state. Now we come to the modeling part of these metabolic networks. So first is constraint-based modeling. So uh, constraint-based modeling approach is based on the assumption that organisms exist in particular environment that typically have scarce resources. And over time, the probability that the fit will survive is higher compared to the probability of the survival of the less fit uh, as in evolution. To be fit for survival, a myriad of constraints must be satisfied, which limits the range of available phenotypes. So the major components of these constraints are first physiochemical constraints, which are basically mass, energy, momentum conservation, etc. And there can be more as well. Biochemical reactions must result in a negative free energy change to proceed in the forward direction. So the Gibbs free energy, delta G, that has to be negative for it to be proceeded in the forward direction, or it'll proceed backwards, or it'll be just in the equilibrium. Uh, Topobiological constraints, which is basically static hindrances, crowding of molecules inside the cells, three-dimensional constraints. Environmental constraints include nutrient availability, pH, temperature, etc., etc., and regulatory constraints as well, because there is a lot of regulation that goes on inside the cell. Each and every component is regulated. The production of each and every enzyme and protein and everything inside the cell is highly regulated by other things. So that is not a hard constraint, but it's a regulatory constraint. Right. Okay. So there we come to the first important kind of a metabolic model, which is the flux balance analysis. So how does it work? So it starts with a set of metabolic reactions. Just okay. Yeah. So FBA or flux balance analysis works on metabolic networks. So first, you need to build reconstruction of a metabolic network is required, consisting of a list of stoichiometrically balanced biochemical modules or reactions. You curate these metabolic reactions. Sorry. Then you formulate the S matrix, which is the stoichiometric matrix. Then you apply mass balance constraints. In this case, this is a steady state condition, which is S dot V equal to 0. Then you define the objective function. So basically, a set of objective function which is given by z equal to ct into v, where basically c is the vector of weights, indicating how much each reaction contributes to the objective function. In practice, when only one reaction is desired for maximization or minimization, c is a vector of zeros, and only one at the position of the particular reaction that you are interested in. Uh, when simulating load, the objective function will have a 1 at the position of the biomass reaction. And finally, you can use linear programming to identify particular flux distributions that maximizes or minimizes this objective function while observing the constraints imposed by mass balance equations and reaction balance. So this is the workflow. You start with the set of reactions in a network. You form the stoichiometric matrix. Then you give the steady state condition is equal to 0. You define an objective function. Uh, and then you optimize the objective function using linear programming, and you find the perfect solution for this particular case. Yeah, again, we've discussed this earlier, but the objective function is actually the most important part of this whole thing, because uh, uh, the FPA's model's objective is to alter the distribution of optimal fluxes within a metabolic network, influencing damage accumulation and lifespan. Uh, So if you do, so basically this was a study that actually showed what a change of your objective function can do. So simulation results indicate that the choice of objective functions significantly impacts replicative lifespan and generation times. Cells without maximal growth optimization show limited growth, while some failing to divide or doing so infrequently with long generation times, emphasizing the uh, importance of maximal growth in replicative aging. So yeah, that is the idea that your objective function is kind of the important of all, which actually kind of tests the fitness of your solutions. And if your objective function is well defined, you'll get good solutions that actually mimic your biological system. If you do not have a good objective function, or if you change your objective function, the kind of solutions that you get are very different. Right. Okay. So yeah, then we're coming to minimization of metabolic adjustment, MoMA, which is actually a kind of a extended version of FPA. Yeah. All uh, the idea for MoMA is basically that assumption of optimality for a wild type organism may be justifiable. The same argument may not be valid for genetically engineered knockouts that were not exposed to long-term evolutionary pressure. In the basis of this method lays the assumption that after a knockout, metabolic fluxes will undergo minimal changes with respect to the flux configuration of the wild type. So the whole assumption is based on robustness and resilience, that even if you knock out a particular gene or a protein from the network, the the metabolic flux change along the metabolic flux change along the network is minimal. So that is the whole assumption for MoMA, which is minimization of metabolic adjustment, as the name suggests. It uses quadratic programming to identify a point in flux space which is closest to the wild type point, based on a conjecture that the mutant remains initially as close as possible to the wild type optimum in terms of flux values. To predict a metabolic phenotype, it uses a flux vector V in a flux space phi of a mutant with the smallest Euclidean distance. So basically, it measures the Euclidean distance between the wild type and the mutated, and it kind of tries to minimize that distance to get that optimal solution based on the assumption that your knockout or your mutant will be as close as possible to your wild type in terms of the metabolic flux. Yeah, this is a good example for that. So if you consider FPA, so for FPA the optimal, so basically here you have two different colors you can see, green and yellow. So I think mm, green is the wild type uh, solution space, and yellow is the knockout solution space. So for FPA, if you use FPA predicts this particular point as the wild type optimal, and if you consider a knockout, FPA predicts this particular point as the FPA of knockout. Right now, if you kind of consider MoMA, so how the change in results is showing is basically that it is not kind of looking for anything more, but it is trying to minimize the distance between the wild type and the knockout. So basically, this is the lowest distance between the wild type and the knockout. So MoMA will give this as an output compared to FPA, which is giving it here, right? So this is a good example of how the results are differing in terms of solution spaces. While on one hand, here is also a very good condition. So if your wild type is here, your knockout should be here. If your wild type is here, knockout should be here, something like this. So basically, you're trying to minimize the flux change between the wild type and the knockout. Okay. Another kind of a measure, or excuse me, sorry. Yeah. Another kind of a model for metabolic networks is ROOM, which is regulatory on-off minimization. The idea here is it involves regulatory constraints in its model for prediction of post-adaptation states. So it minimizes the Euclidean distance between flux distributions of the wild type and the butter strain. MoMA does this, but Room employs a different norm. It focuses on minimizing the total number of significant flux changes, emphasizing the cell's adaptive cost reduction, and it incorporates genetic regulatory changes caused in knockouts, these are minimized by cell to reduce adaptation costs. It operates under the assumption that the regulatory changes can be parsimoniously described by Boolean on-off mechanisms, assigning fixed costs to each regulatory change irrespective of the magnitude. So, yeah. So actually, this is an example of three different models predicting three different outcomes. So yeah, so in this case, your starting is from B1 and your ends at B2 or B3. So the idea is that in a knockout, this particular uh, reaction has been knocked out. C to E has been knocked out. So for MoMA's prediction, it actually uh, yeah, this is actually MoMA's prediction in terms of fluxes. So it predicts 4.4, 4.4, and 8.8 compared to the 10 and 5 when it is just trying to minimize the distance between the difference between the wild type and the knockout. And this is Room's prediction. It finds changes in flux only along a short alternative pathway through V5 and V4. So this is V5, sorry. So this is V5 and V4. So instead of going this way, it is going this way and this way. So uh, it changes flux in only one short alternative pathway, preserving the optimal growth of the wild strain. So it doesn't change any of other of the parameters. It just changes this. So it just incorporates or produces this combination of solutions where you just take this particular reaction route and your flux do not change. So this is actually a good example in terms of the uh, difference in predictions of these three algorithms: FPA, MoMA, and Room. Okay, now we come to gene-protein reaction associations. Uh, Okay. If you have any questions to ask, okay? So I'm just going on and on. So if you have any questions, please stop and ask. Okay. So now we come to gene protein reaction associations. So these are different kind of examples of gene protein reaction associations. So a gene can produce a protein which is involved in a reaction. This is a kind of a simple association where a single gene encodes for a single enzyme, but that might not be a case. So, for example, this is a case of isozymes where multiple genes encode distinct proteins carrying out the same function. So, these proteins are distinct, these genes are also distinct, but their function is same and the enzyme activity is same for the protein. So, these are known as isozymes. The third condition here is a multimeric protein complex. What means is basically a protein is made up of multiple domains, and each of the domain is produced by a separate gene. So these come together and form the active enzyme. So multiple genes are involved in production of one single protein. And last comes the, excuse me, the multifunctional proteins, in which a single protein actually carries out multiple reactions. So one gene producing one protein, and but this protein is actually involved in multiple reactions. Uh, okay, next comes the concept of dead-end metabolites. This also we discussed earlier. So basically, in the whole network, uh, it is a metabolite that is produced by the known metabolic reactions of an organisms, but it has no reactions consuming it. So, for example, in this case, if you forget, so for example, it starts from here, the blue element comes in and it produces the green element. But if you consider these two elements, C and D, and even A, you do not know the origin of A and C. So these are like root non-produced. You do not know the origin of the production of these two. And then you have D, you do not know where it is and how it is getting consumed. So that is known as root non-consumed. These are examples of dead-end metabolites. Uh, in some cases, these dead-end metabolites actually reflect a deficit or an error in how a metabolic database represents knowledge from scientific literature, and it requires further curation of the database. And in other cases, this is actually in this systems level analysis alerts us to areas where more experimental research is required as well, because we do not know the known unknowns of metabolism. So, yeah. Okay, here is the it's the continuing concept of blocked reaction. So if you have dead-end metabolites inside reactions, you do not know from after it has reached the dead-end metabolite what happens with it. So these uh, create this blocked reactions, and which can create issues in the modeling technique. Okay, next comes the concept of flux variability analysis. So it's another modification of flux balance analysis. In case of a flux variability analysis, uh, it quantifies the feasible ranges of reaction fluxes at optimal and suboptimal production rates, and that is determined by FPA. And the flux variability analysis is a generalization of the FPA network, and it aids in understanding of the metabolic flexibility. Uh, Yeah, the idea is to first identify the maximum value of the objective function and constrain the objective function to this value. Then you minimize and maximize each flux independently to identify flexibility in the fluxes across the alternate optima. The idea is for this method is to find how much flexibility is possible between the fluxes of these metabolic networks. So if you have n fluxes, you have 2n FPA problems, something of this order. Yeah, so this, yeah, I'll skip this. It's something that we have already discussed right now. So you have a maximum possible value and a minimum possible value, and this is your range. And your objective is to maximize the VJ, basically the reaction rate, subject to these conditions. Right? So this is how the models work. Okay, so I think, yeah, I'll go to synthetic lethals directly. Okay, so synthetic lethality, what it means is basically um, when inhibitions of two genes or like multiple genes together, it uh, is lethal. Is a lethal phenotype when inhibition of either of the gene is not. So together they are, if you knock them out, they're lethal. But individually, if you knock them out one gene at one time, these are actually not lethal. This is viable. This is actually a very important uh, concept, because if you can do this, you can like if you can find synthetic lethal pairs, it's very easy to kill off the cell or a disease cell or so on. Right? So Classical synthetic lethality occurs when inhibition of two genes is lethal, while inhibition of a single gene is not. It can be harnessed to treat cancer selectively by selecting inactive genes and targeting their synthetic lethal partners. In accordance to this concept, synthetic lethal can also be applied to genes and small molecules, used to elucidate the mode of action of drugs. Cancer is driven by consequences of mutation or amplification of genes that allow cells to escape mechanisms that control proliferation and survival. Hence, inhibitors that target synthetic lethal partners of mutated or amplified genes in cancer cells can cause cancer cell death without affecting the survival of normal cells. So that is very important. So that you can selectively find this synthetic lethal pairs in cancer cells, and you can selectively go and inactivate these genes, and those particular cells will only be hampered and be dead, and it will not affect other cells in that process. Right. So then we come to the concept.
So both need to be inactivated, or inactivating one only
No, no,
can kill?
that is the concept. So inactivating one will not do anything. Uh, but when you inactivate two together, it actually becomes lethal. So it's here. So classical synthetic lethality occurs when inhibition of two genes is lethal, while inhibition of either gene alone is not. Right. So this is the important part. So if you inhibit the single gene, it will not do anything. But if you do it both together, it will be lethal.
Thank you.
Okay. Yeah. So I think I have messed up the slides a little bit. These two should be earlier, but anyway, let's talk about it. RFPA is basically incorporating regulatory constraints into flux balance analysis. Uh, it computes regulatory states consistent with the previous metabolic steady state, utilizes FPA to find flux distributions consistent with current regulatory states. It incorporates regulatory networks in the models. So this is a very good schematic for representation of an integrated metabolic and regulatory network. The regulatory network component consists of set of interactions between the set of transcription factors and genes between themselves and between genes of others. So, for example, this particular transcription factor is inhibiting gene 4, but is overexpressing transcription factor 2, which is actually producing gene 3. So, there's a complex um, uh, relationship between these regulatory elements, but you can incorporate all these in RFPA. The metabolic network component consists of a set of biochemical reactions between metabolites and metabolites available from growth medium as input, and a pseudo-metabolite representing biomass production as input. The regulatory component affects the metabolic component through the expression of proteins that catalyze the biochemical reactions. So basically, what it is saying is this is the metabolic component where you have all the metabolic um, compounds that are involved in the metabolism, but is the whole thing is actually like controlled or regulated by this regulatory network of transcription factors and genes. And if you incorporate these things in your model becomes much more comprehensive and robust. Yeah. So the idea is, uh, yeah, it's similar type of loop, a kind of an iterative process that we discussed earlier. So you start with a set of initial conditions, you derive system states, internal and external concentrations, and then you kind of make the ODE model or the Boolean model. Then you include constraints. Then you do an FP optimization. Then you do system momentum flux, update the system momentum flux values, and kind of use experimental data to kind of validate this and uh, continue iterating unless and until you find a particular good result. I mean, a result that is matching your experimental data or the parameter that is actually matching your known data. Okay, so yeah, now we come to the last part, which are logical models. So we talked about different types of like ODE models. We talked about models like FPA. This is the one of the simpler ones, which are logical models. So for example, in Boolean models, you only have 0 and 1. Either it is inactivated or it is not inactivated, or either it is produced or not. So, but this actually becomes a little bit of an issue if you apply, no, like it's not an issue, but you have to keep certain things in mind because the assumption is either 0 or 1. The thing is, your system can also be somewhere in between. So that is completely possible. So this actually is kind of a small generalization of that virtue, right? So it's the most basic and simplest modeling methodology, and uh, logical models represent the local state of each entity in the system. So, for example, genes, proteins, small molecules at any time level, at discrete level, and the temporal development of the system is often assumed to occur in synchronous discrete time steps. This is another thing. Like you do not have the luxury of using DXDT and integrate over time, because this you're considering time steps, and according to these time steps, your metabolic network and metabolites change from 0 to 1 or 1 to 0, depending on what condition you've put. So the time steps condition is important. So this is a Boolean network. So you have, so for example, this is how the truth table looks like. So basically, in the absence of A and C, B is not present. If C is present, B is present. If A is present, B is not present. If A and C are both present, B is present. So these are different conditions that is describing the relationship between A and C. Similarly, for B and A, and you can get the whole network out of this. Like, so this is much more simpler in terms of like understanding how the model is working. Okay, Boolean models uh, are basically Boolean network models are common qualitative model frameworks for gene regulatory networks. They're capable of describing various biological phenomena such as oscillations, multistationary events, switch-like behavior, and hence these are powerful dynamics for studying GRN dynamics, right? Gene regulatory network. So a lot of papers it's studied on gene regulatory networks using these kind of techniques have used Boolean models only, because that becomes actually a very useful tool, easy and useful tool to study that. Okay, so okay, yeah. Finally, talking about robustness in biological systems. So it is basically we've discussed this earlier as well. So it's considered to be a fundamental feature, and uh, the idea is that if you move the system away from a steady state slightly, does the system come back to its steady state, or does it move to another state? So this representation actually kind of summarizes it in one go. So the state of a system can be showed as a point in the system space. In this case, the state space is simplified into two dimensions. Perturbations forcefully move the point representing the system state. So, for example, uh, this. Uh, so, so this is your. I think this. Yeah. So basically, what is happening here is you have this is your initial steady state at the middle, and then you're perturbing it away from the middle, somewhere here, somewhere here, somewhere here, and here. If you perturb it here, it becomes unstable and of moves out and moves to another state. If you pull it here, this actually pulls back to its steady state. So this is actually showing robustness here. But if you move it here and here, it actually goes to another. So this is a different, these and these are different steady states compared to this. So if you move it farther along, it actually moves to another steady state, right? Yeah, the state of the system might return to its original attractor by adapting to perturbations. There are basics of attractions in the steady state within which the state of the system moves back to the attractor. If the boundary is exceeded, the system might move into an unstable region or move to other attractors as well. As I was saying, so if you move it here, it like is going back to its own original attractor. But if you move it further, here it becomes unstable and moves away. If you move it here, it goes to another steady state. If you move it here, it goes to another steady state. Right? That is all.
So if it is in boundary, then it will move back to the original state. If it
Ah,
is out of boundary, then it will be unstable.
yes, yes. So this is actually the whole system. The whole thing is a system, and you can divide it into like four steady state wala circles. If you move the compound or the whole network out of the steady state of one attractor. So this is one attractor. If you move it out here, in this case, here is no steady state to attract it, so it becomes unstable. In the other cases, there are other steady states that are attracting it better than the original one, so it moves to the other ones. That's
Okay. Okay.
yeah. Uh, Yeah, so now the concept of redundancy and diversity. So yeah, the concept of alternate or fail-safe mechanisms actually enhance this robustness by providing multiple means to achieve a specific function. So it's the same concept that we were discussing a few minutes earlier between multiple edges between two nodes. That is what redundancy is. So you have multiple paths traveling from one node to another, and if you even if you break off one of them, you have two other paths are there to uh, help you out. Right? This mechanism encompasses redundancy, diversity, and overlapping functions, offering flexibility and adaptability. One very good example is gene duplication, followed by specialization and extensive gene loss, serves as a crucial mechanism for evolutionary innovation. So, what happens is if you have a duplicated gene somewhere, it may cause a disease, but if you delete that one gene, one of the duplicated gene, the other gene is still there to take up its job. So it is the redundancy or the fail-safe option there. Uh, Although redundancy is common in duplicated genes, duplicated circuits are rarely observed, suggesting that similar network topologies result from convergent solutions rather than duplication. So the idea is that you can have duplicated genes, but you cannot have like duplicated circuits, duplicated networks. So the whole network cannot be duplicated like that. So somewhere or the other, if you delete a gene, even if that is duplicated, it will fail because the whole network is not duplicated, right? Yeah, so system control is vital to ensure robustness, maintain regulatory feedback, and orchestrating switches between alternative mechanisms to preserve system behavior. And here is an example in budding yeast. These two genes, CLB5 and CLB6, they encode a type of cycling with overlapping functions in cell cycle entry, illustrating redundancy through gene duplication. However, duplicated circuits are not reported, indicating convergent solutions rather than duplication. Phenotypic plasticity, such as shift in yeast metabolism. So basically, dioxic shift is basically a shift in the primary carbon source from, I think, if I'm not lactose to glucose, if I'm not wrong, but like there's a shift in um, uh, the carbon source of the yeast. So I'm not sure from what to what, but there's a shift. That is what is known as a dioxic shift in yeast, and it further contributes to robustness by allowing adaptation to changing environment. So what happens is if you remove a particular the particular carbon source uh, uh, and you put another carbon source in it, the cell still lives because it kind of uses the alternative or the fail-safe option to metabolize the other carbon source compared to the one that it is used to.
But sometimes gene duplication is lethal, no, or it can result in disease
Mhm.
state.
Yeah, yeah. So for example, Huntington's disease is one such case where basically a part of the genome is duplicated, where and you generate a disease. But the idea is that in most cases, that is what it is telling here, that in most cases, the circuits will not be duplicated. One gene might be duplicated, but it's not a very good idea to um, kind of base your whole robustness on duplication of one gene because it is not a straightforward thing. If you duplicate, then it is robust. It does not work like that because you can have multiple other phenotypes and disease states. So the idea is to have alternate or fail-safe options, but duplication is one of them, but it's not a very good option. That is what it is trying to say here, that the circuits are not duplicated. You can have duplicated genes, but if you do not have duplicated circuits, the whole function actually does not change much, right? So it 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 actually is not a very good idea to have it. But in case of this yeast, these are actually same genes, CLB5 and CLB6. Actually, these are same genes, but they are actually uh, producing different types of. So in this case, it is encoding B type cyclins with overlapping functions. So that is a more that is a better idea. So you have slight changes in the gene, but the overlapping cyclins are similar. The B type cyclins that it is producing is similar. This is a better instance, for example. But in case of, say, for example, diaxic shift, you need a fail-safe option. So, for example, not the same gene duplication. So, assume that you are using glucose as your primary source of metabolism, carbon source. Now, when glucose is not available, you have to use something else, say, for example, galactose. Now, your fail-safe option is you turn off your glucose network and you turn on your galactose network. It's not exactly a duplication, but there is a fail-safe option. So duplication is not always a very good idea, but having a fail-safe or an alternative option is always a good idea. Right?
So basically, the circuit should be maintained and
Yeah,
it should not be perturbed.
yeah, that is the idea.
Okay.
Okay, and yeah, finally, we talked about modularity in terms of graph theory, but modularity in biological systems means that you have modules that are specialized. Meaning basically you have divided up the whole network into modules which uh, actually serve as an effective mechanism for containing perturbations and minimizing their effects on the whole system. So the idea is, if you perturb one module, the other modules should not get affected. So if you divide your network into these groups of modules, if you disturb one module, that module should be able to contain it. It should not be spreading to other modules, the perturbation, right? So you can actually restrict the perturbation to a very like, say, for example, if in the metabolic network, one of the elements of your Krebs cycle has been mutated. You do not want that mutation to somehow affect the say, for example, gluconeogenesis, formation of glucose inside the cell. Now, for that to happen, the compartmentalization is important, that you have a separate compartment for, not a separate compartment physically per se, but a separate module of genes and proteins involved in Krebs cycle and a separate modules of genes and proteins involved in gluconeogenesis. So the idea is to have, if you have distinct modules like these, so effect of perturbation in one module uh, does not go on and perturb the other module. So that is the modularity idea. For decoupling, the idea is basically you can have low level you can isolate the low level variations from high level functionalities. So basically, if you have if so it's a bottom up approach. So you have various elements at your low level, various compounds at your low level, and you have a certain functionality at your high level. So for example, the functionality you are talking about is movement of a hand. For this movement of a hand, there are a lot of compounds involved. There are the muscles involved, the actin molecules, the myosin molecules, the other signaling molecules, everything is involved there. So the idea is that a lot of components are involved at the low level, but variations in the components at this low level should not actually uh, perturb the high level functionality, which is folding of the hand. So even if my actin is not correctly, very correctly working, it should not overall hamper my folding of or movement of the hand. So that is the idea. So example is basically molecular chaperones like HSP90, heat shock protein, which not only corrects misfolded proteins but also buffer genetic variations, allowing for adaptation while maintaining genetic diversity. Feedback controls seen in Drosophila segmentation compensate for changes in network interactions or initial states, mitigating impact of mutation. So basically, these are also mechanisms that can actually counteract the mutations, counteract the perturbations in the network. And bistability created through positive feedback can decouple molecular fluctuations from system's committed state, enhancing robustness to genetic and environmental perturbations. So, yeah, that is all. from my side. But the idea is to kind of have fail-safe options and also have mechanisms where the low-level variations do not actually affect your high-level functionalities. That is the idea for robustness and resilience of a network. Okay, so do you have any questions?
No, sir, nice explanation.
Sorry?
No, I don't have any questions. You have explained nicely.
Okay, so if you have any questions before you said it was like 4th May or something, no? Exam?
Yeah, expected 4th May, around 4th May.
Okay. If you have any questions or face any difficulties before that, you can email me here. I'll try to reply and get back to you. And yeah, other than that, all the best for your exam. And thank you for joining. Thank you for doing this
Thank you.
course.
Thank you.
Hope you have liked the
So please
course.
upload this session as well.
Yeah, I'll upload this as well. Yesterday's one has already been uploaded, so you can check that out.
Yeah,
This
previous
one,
one is uploaded.
this one also I'll upload.
Yeah.
And I'll not
Okay.
upload the PPT separately because it is already in the previous week's uh,
Okay.
PPT only. If you
Okay.
open week 12, this part is all already there.
Okay. Okay.
Okay.
Okay.
Okay.
Thank you.
Thank you. Thank you for joining. Yeah.
Thank you.